So in this paper, what they were basically able to show was that when you compare the amount of upregulated and downregulated genes in the amygdala of these mice, when you look at the number of genes spent just during naive behavior versus social interaction, specifically the germ-free mice have a huge increase in different genes that are activated throughout, showing kind of atypical brain patterns. And of these genes, the ones that show the biggest differences are actually MAP kinase signaling pathways and spliceosomes. So this is a figure just kind of depicting what we were kind of visually looking at earlier. The CC group is the conventional controls. The red bar is how much time they spend, sorry, the red bar here is how much time they spend with the novel conspecific, and the blue is how much time they spend with the familiar, and the y-axis is just the time. And basically what you expect in a normal mouse is that they spend much more time with the novel mouse rather than the familiar. And interestingly, in the germ-free model, this effect is lost. They can't really distinguish between novel and familiar, or show that there's a preference in interacting with the novel over the familiar. And what the GFC here stands for is germ-free colonized or conventionalized. That's where a microbiome is added back to the germ-free mouse. And in this case, it slightly improves it, but not significantly. And of course, what we are always trying to do is show what this can mean translationally. So another model that we often study here is that of the cesarean section birth, just because different birth mode types can actually have large differences on the microbiome as a whole. And a lot of this is because rather than getting the first seeding from birth of the maternal microbiome by C-section, the infant's microbiome will actually look more closely to the skin microbiome of the mother rather than the gut. So we use the C-section models to test different aspects of behavior. And interestingly, what we've seen is, again, same what we normally see in controls, where they spend more time with the novel conspecific rather than the familiar. And then in the cross-foster group, so just swapping litters as a control group, this effect is still the same. But when a C-section is performed, this effect is lost. So it's kind of interesting that it's showing similar response as what we see in the germ-free group of how the microbiome can actually alter the social behavior of the host. And what's interesting too is when we look at the effects of different antibiotics in this test. And generally, again, we have just the vehicle control. You see that there's a novel preference, but then when penicillin V is given, there's actually a reduction in this preference for the novel conspecific. So it's kind of resembling what we see both in the germ-free and the C-section models. But interestingly, it's not always the case, because sometimes, in this case, an antibiotic cocktail completely abolished what we saw in the C-section effect. So it can go on to say that it could be specific microbes and combination of antibiotics that can be used that may have different effects. But it's kind of just an example that it is a very complex story, and we're still unraveling a lot of it as we go. A lot of this is summarized really nicely in this paper that was published a couple of years ago by Livia Moret, basically looking at the different effects of behavior that happens as a result of C-section, from sociability to anxiety-like behavior to repetitive behaviors, and how this can actually be restored or fixed by using a series of different probiotics, which are live bacteria used to confer a health benefit. In this case, it was Bifidobacterium species. So that kind of takes me on to the next part of the talk that I want to bring up. And it starts kind of with the critical windows for neuroplasticity, which is a good thing to kind of be aware of when we're looking at different aspects of health and development. And that is just that in different stages of life, we have different kind of critical windows that different diseases or disorders may occur. And this could be due to a variety of factors, not only the microbiome, but it's something that we're aware of and we're studying as we go. So for example, at birth, you could have things like sensory function being impaired if something were to occur. And as you go on, there are different orders of cognition that could potentially be affected. And then there are some diseases and disorders that start to take place during these times, from autism spectrum disorder to ADHD or even schizophrenia later in adolescence and adulthood. So with autism, it's actually a pretty interesting set of conditions that we try to explore as much as we can in the context of the microbiome. It's a condition that's characterized by having a high degree of heritability as far as neurodevelopmental disorders go. It is fairly common in the general population, more common in males than females. And the three symptom domains that really appear to translate across different species as well is abnormal social interaction, impaired communication, and repetitive behaviors. And what's really interesting about this is that there are differences found in a variety of papers at this point, looking at the microbiome and individuals with autism spectrum disorder. And very interestingly as well is that we see an increase in widespread RNA editing, similar to that of what we see in germ-free individuals. All just kind of little hints and clues that the microbiome could very well be a contributing factor to parts of this spectrum disorder. So there are a number of different papers that have been coming out that use animal models to study autism more closely, all looking at different parts, from bile acid and tryptophan metabolism to use of probiotics to try and treat some of these different effects. And fortunately, probiotics have shown some benefits when looking at things like different social behaviors. So the goal is to be able to find different probiotics that might be able to culminate into us having a series of potential treatments and personalized treatments for different individuals. And that takes me to kind of another aspect of the talk that I wanted to share. That's fecal microbiota transplantation, or FMT. You might have heard of this. This is something that is first kind of been reintroduced by treatment for Clostridium difficile for nosocomial hospital infections of recurrence, C. diff. And what's really amazing is this has shown a higher degree of efficacy than even the top line antibiotics, with FMT being able to prevent recurrent Clostridium difficile from occurring. The last studies I saw were greater than 90%, which is extremely impressive. But what's really interesting is this isn't a new technique. In fact, there are records showing from Gehong that ancient Chinese researcher gave a yellow soup to treat his patients with severe diarrhea, which on the one hand, when someone is extremely sick, you would be willing to do just about anything. But I'm glad to say that that is not exactly the treatment that we look to nowadays. There are different pills, sometimes referred to as capsules, that have been used for basically giving these FMTs, and sometimes either direction, depending on what you're targeting. And what's really interesting is that by using FMT, there has been a few studies that have gone back and shown that they're able to improve things like the gastrointestinal and autism symptoms, which is really interesting and kind of showing that we are going along the right path, that there are a lot of systems biology approaches that are good to be aware of, and that the microbiome could play a very important role in this disorder. More support for this actually came out in a publication a couple of years ago by Sarkis Masmanian's group down there closer to you in Caltech, where they actually transferred a range of donors who have autism spectrum as well as healthy controls to germ-free mice, and then categorize their behavior, and then went further to show that certain things can be improved depending on the treatments. But this type of approach needs to be used very carefully, because there are a lot of people out in the field, and how things are reported on in the news can often stretch what is being produced in the lab. So it's just good to look firsthand at the research and not always just believe what is being tweeted or brought up in the more mainstream media. But there's also a lot of potential for other social disorders being explored further. Schizophrenia being one, and there's already been some work showing that there is differences in the microbiota, as well as bipolar disorder, and also something that we're working on here, something I've personally been working on, in fact, social anxiety disorder. So earlier this year, we actually published a paper showing that there was differences in the microbiome and social anxiety disorder, which is characterized by individuals who have an extreme reaction to different social settings and basically not being able to function as what we would consider a normal life because of these extreme inhibitions and not being able to go and hold a normal job or do things as simple as going to a restaurant or going to the grocery store. So it can really be difficult on these individuals, which is one of the reasons we want to kind of categorize it further. So there's actually going to be another study coming out soon. We're hoping to get it published in the next few months, basically where we have done something similar to what the Macedonian group did, where we're trying to create a translational model of social anxiety disorder. So we actually recruited patients that were included in the previous study, and we prepped their stool for fecal microbiome transplant and selected specific donors who were screened for having different microbiota differences, as shown here in a PCA plot of beta diversity, the healthy controls in the blue and the social anxiety in red. And with the little marking on them are each one of our donors. We also categorize them dependent on their Leibowitz social anxiety scores, their body mass index, and then gender balance fit. And then we transferred those FMTs to their own individual donor groups of 6 mice so that we can then compare them and see if they show differences and if we can further categorize it. I'm excited to say that we do see some interesting results, saying that social fear might be something that can be transferred across species due to the microbiome. However, it's not a total explanation of social fear, but in some cases it might be something that could be something that can lead to it or exacerbate the symptoms. So to kind of just recap what the microbiota gut-brain axis and social and its effects on social behavior, is that there are a number of different aspects of daily life that can be affected by both the microbiota and the gut and the brain. And there are a series of different influencing factors that we can have control over. And through this, we can actually affect downstream effects on life that can hopefully improve things like empathy and altruism and trust across different social groups.